Good day, Homo sapiens. How are you doing? And welcome back to studying the Prescott's Microbiology. And we are in Chapter 2. Chapter 2 is about microscopes and the specimens that we will be studying. How to prepare them and how to study them under microscope, all of the staining techniques and how to smear them and all of that, and what are the types of general microscope. Oh guys, wait, give me a second. I just wanted to check if it will work fine. Right, as long as it works fine. This is my daily routine to check it, because I have went through a lot when I didn't check right. So what I was saying, that we will be studying microscope, and we already studied the types of the light microscope, and only one type of microscope, the type we studied. Sorry for my slow wording and jumbling. I'm slow at start 2 minutes or 3 minutes, so yeah. Let me breathe and say it again. Okay, so we studied how the lenses work, right? The lenses are the important part of the microscope that we use, because lenses are the part of the microscope that actually do all the working of magnification of the specimens that we study under the microscope. So we studied how the lenses work, right? And we use the convex lens, and only we study the refraction, right? And we don't study the reflection or the diffraction. And in bright field microscope, this is the one type of the light microscope, and that is the background is white. When you study the specimen is colored, but the background is white. So we were just studying how the bright field microscope was constructed, and this is how it was constructed, and we studied all of that. Today we are going to study an important topic, and that is the microscope resolution. Like guys, how does the resolution of the microscope works? Resolution, we know how the lenses are fitted and how actually does the lens like focal length or these kind of things work, but we don't actually know how actually the resolution can work, right? So we study that first before going into the other types of the microscope, that are the dark field microscope. Where is it? Here, the dark field microscope, the phase contrast microscope, and yeah, I think that's it for the types. Yeah, the fluorescence microscope. Alright, so let's go back, guys. Finally we are starting. Are you guys ready? I don't want to let me enlarge in it so we can see it clearly. If there is any problem with it, tell me. Like I told you, I am start at 2 minutes. Once I start, I will finish my blabbering, right? So microscope resolution. What is resolution first? So the most important part of the microscope is the objective, which must produce a clear image, not just a magnified one, right? It shouldn't just enlarge it should be clear to be able to be seen. Thus resolution is extremely important. One is magnification, one is resolution. Magnification is just enlarging the size. Resolution is the ability of a lens to separate or distinguish between small objects that are close together. Like they should be distinct, they should be clear apart, and there shouldn't be no boundaries or blurry boundaries. There should be clear-cut boundaries. Resolution is described mathematically by an equation developed in 1870 by Ernst Abbe. I don't know what this word is. A German physicist responsible for much of the optical theory underlying in the microscope design. Optical theory, responsible for much of the optical theory underlying the microscope. All right. So this German scientist was actually the one that was actually explaining the work of the microscope, or designed a microscope, like the optical working of the microscope. I think it was enhanced by the scientist. We'll study more. What did he do? The Abbe equation states the minimal distance d between two objects that reveals them as separate entities depends on the wavelength line used to illuminate the specimen and on the numerical aperture of the lens, which is the ability of the lens to gather the light. So this scientist, he actually found out the distance which is needed, right, for the lens and the light. Like for that distance, for you can say this is a focal length, right? Like he was discovering what length would be the best for the specimens to be distant from the lens to for the specimen to be distant from the lens to work perfectly to capture, in the lenses the light is focused more like this, all right? So he was actually measuring the distance, what distance would be the best for the lens to work perfectly, to point out that light on the specimen perfectly. He distanced that light, that D, and the wavelength of the light. Guys, wavelength of the light is important because every wavelength of the light carries different energy, like you can say, and it has different frequency of that. If it has different frequency, means it has different speed and it has different energy. So it depends, because we want to know what amount of energy we want to. Usually the higher the smaller the wavelength, the higher the energy is, and the better it is for us to see the specimen. To illuminate the specimen and the numerical aperture of the lens. Guys, this is which is the ability of lens to gather the light. All right, he told us. He said that how does it study the D and this lambda? It was on the numerical aperture of the lens. Yeah, it is all related to the lens, and that is n sine theta, which is the ability of the lens to gather light. I didn't understood a single word. Should I read more further? This D, this wavelength, and this n sine theta, numerical aperture, I'm not getting a single word. Wait, let me read further. As D becomes smaller, the resolution increases and the finer detail can be discerned in a specimen. So they are saying the more closer it is, the more the resolution increases, and it only means the lesser the focal length. Guys, we previously studied the smaller the focal length is, the better it is for the lens to work, right? So the same case is this D. I think this D is referring to the focal length of the lenses. So they are saying the smaller it is, the resolution increases and finer detail can be discerned in a specimen. D becomes small as the wavelength of the light used decreases, right? When did it become smaller? It's not it's not just the ability of the lens to have a smaller focal length. It also depends on the wavelength of the light that is used, right? The wavelength of the light confirms, or you can say it depends on the wavelength of the light, how it is bent in a medium, right? How the refraction works. Refraction works by bending the light. So if the light has different wavelengths, it is going to be bent differently than one another. So they are saying the more the wavelength is smaller, the more the D becomes smaller, means the more the bending occurs, the higher bending occurs. So it makes the D smaller. And wait, should I show you in the diagram, or pencil here? Let me draw it a little bit. Guys, if you are thinking I will be doing things fastly, you are getting it wrong, because I want to enjoy it studying, so I'm not going fast or skipping things. So guys, this is the length. Wait, I wanna draw. So this is the length, this is the specimen, the lens focal length. All right, this is not funny. What did I draw? So this is the lens, this is the object, right? The lens usually has a focal length. The smaller the focal length is, the better. The higher the focal length is, the bad the resolution is, right? The lens of focal length of the lens. While it also depends on the wavelength. So either the light has this wavelength, right, or either it has, you know, smaller wavelength. You get it what I'm saying, right? Wavelength is this, the difference, or the distance between two crusts, or the difference between two troughs, right? So we need this smaller wavelength. If it is, you know, the larger wavelength, it's going to be bend less. Also, the focal length works by pointing the light, you know, like focusing it on one point, right? Usually the convex lens works this way, right? So, and what is bent here, this is the light bending, right? So if we have the larger wavelength and if we have smaller wavelengths, let's draw it different separately. Here, this is one lens, this is another lens. This one has larger wavelength going through it, this one has smaller wavelength, right? Means higher frequency. So this is going to bend the light more sharply, while this one is going to not bend the light as sharply, as you can see, right? It's going to bend. So it means like it reaches the point early than this one. I don't know if you guys are understanding or not, but the more it bends sharply, the lesser the distance becomes. Like the more it points towards it. While if you look at in this case, it needs this much distance for it to cover, means the focal length increases automatically. So let's move on further. And as the numerical aperture increases, guys, I still don't get it. What is this numerical aperture, guys? I need to go to Google for this. Numerical aperture or whatever. What is numerical aperture of lens? I know it will be some easy definition, but what can I do if I'm not? Is a measurement of its ability to gather light, essentially representing the range of the angles at which the light can enter the lens. And it is calculated by multiplying the refractive index of the surrounding medium by sine of half of cone angle of the light entering the lens. A higher NA indicates the greater ability to capture the light and resolve finer details, making crucial for microscopy. Is there high numerical aperture versus low? Let me see it in the diagrams, if you know. What is this? How do we know which one works fine? The smaller the aperture, which one is smaller? Here, 0.12, 0.34, 0.87. The A has the smallest. All right. I see. Alright, so as you can see, the first one, the smaller the NA, the numerical aperture was. No! Didn't it need it to be high? It needs to be high. A high number indicates the lens can capture. Yeah, it needs to be high. So we need the highest one, and that is 0.12. Oh, sorry guys. Yeah, 0.12 is the largest number, 0.87 is the smallest number. As you can see, the 0.87 gathered this less light, and as you can see, you know, the distance is way too small and the light is bent, you know, very sharply, but you can see the light is very less here. While in this one, 0.12, this is the largest one, and the numerical aperture is high. I think I kind of get the gist of it. So, the D becomes smaller as the wavelength of the light used decreases, as the numerical aperture increases. Alright, so if the D becomes smaller, the numerical aperture becomes larger. And they are saying the larger it is, the more the light is captured and bent. Right. Did you get something? Thus, the greatest resolution is obtained using a lens with the largest possible NA. Okay, the larger it is, the better it is. And the light of the shortest wavelength, and the wavelength should be short as possible. At the blue end of the visible spectrum in the range of 450 to 550 nanometer. See figure 6.25? 6.25? Do I know what 6.25? Where it is? Figure 6.25. Where is 6.25? Whatever, I'm not looking at 6.25. Oh, this is talking about what? Chapter 6 or something? I'm not going there. We are going to Google for this. So, all the wavelengths of the light. Wavelengths of the light, right. So blurry images, I don't know if we can see it here. Here, I think this one is pretty normal one a little bit. So as you can see, the visible light is the white light, which we usually use, right, for illumination purposes. So it starts from red and ends on the violet or blue, you know. So the red has the largest wavelength, mean it has this lower energy, right? So 700 nanometer. That you have to know what the wavelength is, okay? The distance between two crusts or the distance between two troughs. You know the highest points of the wavelength, you know it's a curve, so the curves. So they are talking about the curves distance. So 700 nanometer and the 400 nanometer. 400 nanometer is the lower one. So the lower the wavelength is, the better it is. And we don't have to go look at the C figure. So they are saying 450 to 500 nanometer, that is the blue end of the, like it is starting from blue. Does it start from blue? Am I tripping? 450 to 500. Yeah, they did mention 500. So yeah, it's starting from 500. 450 to 500. Yeah, from the opposite side to the inner side. Yeah. So the numerical aperture. Now, what the heck? Now they are explaining what the numerical aperture is. They should have explained it more first. Whatever. N sin theta of a lens is a complex concept that can be difficult to understand. Alright, so you explain it. It is defined by two components. N is the refractive index of the medium in which the lens works. That is air, right? In the microscope, it is air. And theta is the 1/2 angle of the cone of light entering an object. This is lens actually, right? This objective word which is used here, it is the lens, objective lens. So this is the 1/2. You mean if the light is entering the angle at 100 degrees centigrade, right? Usually it's not 100, but whatever. But this theta is like 550 degrees centigrade, because it is 1/2. If you divide 100 by 2, it becomes 50. The angle of the cone of light entering an object. So we know that when this cone has a narrow angle and tapers to sharper point, it does not spread out much after leaving the slide and therefore does not adequately separate images. Alright, now I get it. When we were looking at the diagram here, as you can see, wait, where did the diagram go? Here, I can explain. So they are saying if the NA, the numerical aperture of the lens, is higher, it is better because why? Because it doesn't spread out after going through the specimen, right? Here, you know, this is the sharper here. Wait, this is actually a pretty good image. So wait, let me finish the previous sentence. That is that it is sharper, right? You can see it is sharper. So when it passes through the object, it doesn't spread out that much, and the object image remains constant. While with this case, this is the smaller one, and I think as soon as this passes through this specimen, it spreads out, making the image doesn't look as neat as, you know, this one, right? So guys, 0.1 to 0.4. Wait, which one is bigger? 0.12 or 0.87. Oh my goodness. Oh my goodness. Guys, can't you even comment? Oh my goodness. 0.12 is smaller. 0.87 is larger. It is closer to 1. Guys, my concept was wrong. So this one is the sharper one. This isn't the sharper one. It has more light and it has, curved it more. It has curved it more and more sharply. So this is going to let the light spread out after it passes through the specimen, while this one isn't going to let. Finally, I get it. The larger the Na number means the smaller the distance will be. Everything is adding up. Look at this distance. This is smaller, while the Na value is larger. The angle is sharper. Well, look at this one. The D just automatically goes long and the number is smaller. The angle is just 7 degrees. You guys should have told me that I was misunderstanding this 0.1 number. I was thinking of positive and negatives. In negative, usually the smaller the number is, the larger it is. The larger the number is, the smaller the value gets in negative. But this isn't negative. This is points, decimals. Oh my gosh. What the heck? Why isn't it moving now? So this was the this was the optical index, half angle, focal length, a lens. So this is lens. It is better if the distance is smaller, and that will only happen if the wavelength of the light is shorter, and automatically the aperture optical index, or you can say numerical aperture, will become larger. That means the distance will become smaller, and as you can see, this is the focal length, or you can call it. Wait a second. Don't tell me I am getting wrong something. I hope now they don't tell me this is the D that they were talking about. I am pretty sure that they were talking about this turbidity. Guys, don't tell me they are talking about this D. I'm going to get angry now because what is this? So they are talking about this D. The distance. The distance is smaller. Objective lens. I need a better diagram for this. Or should we go watch a YouTube video? NA. Let me see, what is this? This is D, NA, sine theta. This should be an numerical aperture. I get it, I get it. I don't have to go through more detail. So, what are they talking about now? So they are saying N is the refractive index of medium in which the lens works. So we measure that. And 0 is the 1/2 angle of the cone of light entering an objective lens. Alright, we get this one as well. So when this cone has a narrow angle and tapers to a sharper point, narrow angle, narrow means it's more sharper angle, it is bent more, as we have seen in the diagram previously, and tapers to a sharper point, of course, a sharper point. It does not spread out much after leaving the slide and therefore does not adequately separate the image. I hope I got the point right, okay? I will get angry if it was wrong. Yeah, they are saying the larger the numerical aperture is, the better it is. And it will clearly seize the closely packed object. If the cone of the light has a wide guide, this thing that we saw, the V shape and a little bit of that lens over up there, they call this cone of light, and has a very wide range angle, very wide angle, spreads out rapidly after passing through the specimen. Yeah, they are saying if the cone has larger angle, right? Like, this thing, like if this is lens, that is the cone, right? And now this is the cone. This is the cone. Like if you look at this, It has, why, wait, I draw the diagram wrong, and now I can't make the angles. Okay, I need to make it look more okay. Whatever, I get what they are saying. So this angle and this angle, while I try to justify it, they are saying the more the cone angle is, no, the more the larger the cone has, it has, you know, more angle, or you can say if it has wide angle, it is going to spread more, and it has to have a larger size, and when it passes through the specimen, it's going to spread, right? While if this, in this case, if the angle is smaller, it doesn't have, it won't be spreading after it passes through the specimen, right? So it won't pass through that much as it this one. So everything will be fine. Alright, so what happens then? The angle of the cone of light that can enter a lens depends on the refractive index of a medium in which the lens works, right? They are saying the cone that is made depends on the refractive index of the medium, right? If it happens, if the light is passing from the solid into air, or into liquid, or again into the solid, as well as upon the objective lens itself. Yeah, they are saying it depends, you know, on the lens as well, and it depends on the medium as well. The refractive index of the air is 1, and the sin theta cannot be greater than 1. Yeah, they are saying that the sin theta cannot be greater than 1. So the maximum theta is 90 degree and sin 90 is 1. That means the angle shouldn't be greater than 90. Guys, this is 90 degree, right? This is cone. I don't know how I made this, whatever, we will just consider it. So this is a 90 degree, like you guys get it, right? It should be 90 degree. If it goes more than 90 degree, it won't make a coney one, so it shouldn't be greater than 90 degree, and that means the refractive index should be 1, should not be greater than 1. Therefore, no lens works in the air have the numerical aperture greater than 1. Yeah, the only practical way to raise the numerical aperture above 1, therefore achieve higher resolution, is to increase the refractive index with the immersion soil, a colorless liquid with the same refractive index as the glass, right? So they are saying that the refractive index of the lens is 1, right? Like they can refract the light only until 90 degree. If it goes maximum above that, it won't work normally. So how do we increase that refractive index from 90 degree and from 1 above 1? How do we do it? We immerse it into a soil. They are saying the oil has the same refractive index as the lens, and if we combine them, it will have glass. They means lens. So it will, if we combine them, it will make the refractive index more. If the air is replaced with the immersion oil, many light rays that did not enter the objective lens due to reflection and the refraction at the surface of the objective lens and slide will now do so. Yeah, we know there is a reflection that occurs through glass. So if you remove the quantity of the reflected light, it will turn into the refracted light as well, and the quantity of the light will increase, and the refraction at the surface of the objective lens and slide will now do so. They are saying that the refraction that were happening beyond 90 degree will now turn into more 90 degrees now, so increasing the light, and you can say increasing the numerical aperture and resolution results. Numerical aperture is related to another characteristics of the objective lens, the working distance. Yeah, they are saying that numerical aperture doesn't just relate to the refractive index or things like that, but another characteristics of the objective lens, and that was the working distance, right? The working distance of an object is the distance between the front surface of the lens and the surface of the cover glass, if one is used, or the specimen when it is in the sharp focus. Guys, this is the lens, this is the specimen, and the D that they are talking about, the working distance, is this. It's this thing is either the cover slip or the specimen, right? And this is the front, means lens is 3D, right? So we are talking about the frontmost part of the lens. They are saying that is the working distance. And the surface of the cover glass, the square rectangular shape that we use, what do you call the slide, and to cover our specimen we use a cover glass, right? So they are talking about that. Objectives with that, guys, they are using the shortcuts. Okay, this is lens with a large numerical aperture and greater resolving power have short working distance, right? Like, short working distance is directly proportional to higher resolution. The higher numerical aperture is directly related to the higher resolution power. So the preceding discussion has focused on the resolving power of the objective lens. The resolution of an entire microscope must take into account the numerical aperture of its condenser, as is evident from the equation below. All right. Oh my god, guys, till now what were we studying? We were talking about the numerical aperture, we were talking about the refractive index, we were talking about the angles, we were talking about the D's. But of what? Only of the objective lens. If you are wanting to know about the resolution of the whole microscope, you have to include the condenser numerical aperture as well. Guys, when the light comes, right, the light is not condensed only through lens. When it happens, there is a quantity of light. Why is it not working, guys? It doesn't work. I don't know why. Right, why? Oh, okay, so that was above the page. This is lens. When the light enters into it, it kind of, you can say, if the light goes here and here, the lens cannot capture it, kind of condenses this kind of light. But there is by the way, light doesn't come from the above, it comes from the below, right? And so usually there is a condenser. You can say the light is coming from below, but there is a condenser as well. If the light is going all the way to here and here, you can say, right, and the specimen is just here, right, while the lens is above that, right here. Why can't I go even more up? You can say the light is up here. So the light that is going to pass through this specimen, right, you can't, what are you going to do with all of this light if it passes through like this, right? So that's why it doesn't work pretty well if we only use the light source. We put a condenser in between, which condenses the light and make it more towards the specimen. So when the light that is passed to the specimen goes directly to the lens and doesn't have extra here and there light passing through it, so it kind of what do you call it, specialize, no, enhance, it kind of enhance the working of the lens. So they are saying we have to take in account the numerical aperture of its condenser as well. And how do we know it happens? D microscope. So this is the distance of the from the microscope source of light, I think. I don't know what does that mean. Yeah, but there we were talking about the D of the objective lens, but here we are talking about the microscope, and that only means, I think, condenser, which is in the stage of the microscope. This lambda again is the lambda of the light, the wavelength of the light, NA objective plus NA. All right, what the heck am I doing? So they are saying if you want to know about the D of the whole microscope, you have to add the numerical aperture of both the objective lens and the condenser. The condenser is a large light gathering lens used to project a wide cone of light through the slide and into the objective lens. Yeah, it is used to project the there was a wide cone of light first, but the condenser narrow is and make it possible for all of it to pass through the specimen, so more light is captured by the lens, and so the clearer the object image is. Most microscopes have a condenser with a numerical aperture between 1.2 to 1.4. Okay, 1.2 to 1.4 is quite high number. However, the condenser numerical aperture will not be much above 0.9 unless the top of the condenser is oiled to the bottom of the slide. Alright. So they are saying that usually microscope have condenser that has 1.2 to 1.4, but they are saying if we use normally, the condenser doesn't have value much above 0.9. So how do we make it reach to 1.2 to and 1.4? They are saying if the condenser is oiled, just as we studied about the objective length increasing in the refractive index, we can do the same with the condenser as well. To the bottom of the slide, right? Bottom of the slide, if the above portion of the condenser is covered, that is the bottom of the slide part. So we need to oil that part, not the oiling of the part that is, not the oiling of the, this is condenser lens. Here we had our slide, so we need to cover this area with the oil, and the light is coming, this is the light source, and light is going, oh sorry, I made it way too big. Like you can see, the light is going from here, right? So we don't have to cover this area with oil, we have to cover this area with oil, which is just below our slide, and the up front part of the our objective condenser lens. So we increase the numerical aperture by that. During routine microscope operation, the condenser usually is not oiled, and this limits the overall resolution even with an oil immersion objective. Yeah, they are saying if you didn't oil the condenser, it limits the overall resolution even if you have oil immersed objective. Means if you want overall resolution, better resolution, you immerse oil on the objective lens as well as the condenser. Although the resolution of the microscope must consider both the condenser and the objective lens, in most cases the limit of resolution of the light microscope is calculated using the Ab's equation. Now we are coming towards Ab. Finally, completely forgot about this guy, which considers the objective lens only. Okay, alright, if we are going to go about the Ab's equation, we are going to study the resolution of light microscope from the objective lens only. The maximum theoretical resolving power of a microscope with an oil immersed objective lens is the numerical aperture of 1.25, and the blue-green light is approximately 0.2 micrometer. Blue-green light, we already know that the limit is from blue light, right? Well, green is just so near to blue, so I think they also calculated the green light with as well, because till now we were studying only about the blue and purple part, right? So they are saying if the theoretical, not the practical, always remember the theoretical value always comes perfect, but in real life, microscope has more problems, air resistance, particles, and much more things than theory. So the practical value usually comes lower than the theoretical value. So the theoretical resolving power of the microscope is 1.25. So D, 0.5 to 530 nanometer. This is the light, right? This is the light wavelength. What is the 0.5? What is the 0.5? Is it the refractive index? What was 1.2? What is this? Did we read this formula somewhere? Alright, so 0.5 is the formula, and that d lambda was the 530 meter, and n sine theta is 1.25. Alright, so the numerical aperture is the 1.25. So if we calculate the resolving power of the light microscope by Ab's equation, this is Ab's equation, which is given below, right? Or which is given above. That is, we get 212 nanometer or 0.2 micrometer resolution. That is the resolving power if we go with the theoretical equation of the abs, right? At best, a bright field microscope can distinguish between two dots about 0.2 micrometer apart. Alright, so it's resolving power. So resolving power is to differentiate between two objects. Like how much closer should be the objects, or you can say how much closer two objects can they differentiate. Like microorganisms are so small and they can be close together, in nanometers. So it depends on the microscope how deeper it can go into that distance. So according to the abs, if we have 0.5, we are talking about the light microscope, because the blue-green light is used with the blue-green light. Alright, so the blue-green light is approximately 0.2 micrometer. Alright. Blue, you know why they showed the blue-green light, right? Usually light microscope this happens, right? And this is also the bright field microscope. In electron microscope, it is a different story, because we don't use light and we actually use the lenses the same way, right? We use the lenses not that much different. Lenses cannot be that much different, like it doesn't. But the whole process changes in the electron microscope, and the resolution power exceeds to nanometer, that much lower. It's because of the electron beams that we use in there. It has approximately like high energy, higher so many times, I don't know how many times more, 100,000 times more energy than these lights, the blue-green lights. Blue-green light is the maximum power energy, wavelength, but it still don't have that much energy as the electron beam. So can distinguish between two dots about 0.2 micrometer, but it's still way too much, because our eyes can differentiate, I think, 1 millimeter or 0.2 millimeter, 1 millimeter or 2 millimeter. We can distinguish two objects apart if only they are 1 millimeter, distant, or 2 millimeter. You can clearly say that when you look at the scale, ruler. If you have a ruler, it has inches, centimeter, and then millimeter. Try to distance the two lines of 1 millimeter. You can do it, because it's just 1 millimeter apart. Like you cannot go beyond that. How are you going to see in the middle of that if they close that distance as well? So our eyes are just in millimeters, while this is in micrometers, right? This is way too big. The same size as the very small bacteria, right? So not the larger bacterium, but the very small bacterium, means it can differentiate up till that smaller of bacteria. So how does it change its weight? Alright, so this, wait, we are not going towards that. Alright, why was I going to Google for everything? I was reading everything while the Google window was covering everything. That is so bad. So here you can look at these, right? Look at this. When I was saying this and that and that. Alright. Why do I do this every time? So this is the objective lens, and this is the working distance. This is the slide with the specimen. This is the theta, and this is the theta. Here, the angular aperture theta is 1/2 angle of the cone of light that enters the lens from a specimen, and the numerical aperture is n sine theta. Alright, so they are talking about the theta of this. While I was studying, when I was showing you guys, I was talking about this angle, which I was completely wrong, it seems like. What theta they are talking about is this theta here in the below. And they say when we talk about theta in the formula, we divide it 1/2, and then we only talk about the half part. Like there are actually two angles in there. So I was wrong. Guys, I'm not a teacher of you guys. I'm reading this book for the first time. So yeah. So this is a study together. So don't expect me I'll be right. I won't be right, because like I said, I am very weak at studying the lenses. I never focused on it. So yeah. So they are talking about this theta. Alright, so that was in that. In the right hand illustration, the lens has a larger angle and the numerical aperture. Its resolution is greater, its working distance smaller. Right, they are saying the bigger the angle is, the better the numerical aperture will be, and that means the distance will be smaller. Alright, while here it is the oil immersion objective. An oil immersion objective operating in the air with immersive oil. Okay, so they put the oil in the middle and then the light when passes through it upper front of the wait, did they put the oil on the glass or did they put oil on the lens? I completely thought that they put oil on the objective. Whatever, it works the same way. The light just doesn't reflect, it doesn't change its trajectory that much because oil kind of captures it, and then it most quantity of the light goes past through it. So I think they put the oil right from the lens to the glass, like it is filling the air, right? Like it doesn't have air, instead of air it have oil, so the refractive index became higher. So I didn't know that they will be changing, or filling the whole air area. I just thought they were putting some oil up there and some oil up there and everything would be fine. No, they put the whole area, put oil in the whole area. Alright. The properties of microscope objective. So property magnification. Guys, I'm not getting what is objective. What is objective? Isn't it the lens? I need to know. What is objective in microscope? I hope it is lens or I'm going to go crazy. Objective is a lens or group of lenses that focuses the light. Alright, I was going to go crazy there. So, magnification 4x, low power 10x, high power 40 to 45x, oil immersion 90 to 90x. This is the scanning and this is the magnification, right? So when it is on low power, means low light. Guys, they have to be more specific. Now they're talking about light here. So it is 10x when it is in best condition, like high power, high light. You can say low wavelength light. So it's 40 to 45x. While in oil immersion, 90 to 100x. I see it doubles. Then the numerical aperture 0.10, 0.25, 0.55. Okay. So if the numerical aperture is 0.25, that means it's going to not work at its best. The numerical aperture is higher, it's going to work best. And the best is 1.25 to 1.4, and that happens with oil immersion. Focal length, the smaller the focal length is, the better it is. As you can see, 40 millimeter is just for scanning, while 16 millimeter you can resolve it better. Low power, and it becomes even higher power with the lower. The lower the focal length is, the better it is. You can see here it becomes even more lower through oil immersion. Working distance, the smaller it is, the better it is, just as with the focal length. While resolving power with a light of 450 nanometer blue light, the lower the wavelength is, the better it is, because the energy is higher in the lower wavelength. So 2.3, 0.9, 0.35, 0.8. As you can see, the numbers, all of these three goes lower, and so the work becomes the best. While in this case, it becomes, when the refractive index or numerical aperture is higher, it works the best. And it all happens through oil immersion. Means oil immersion is just the best savior of everything. Okay, So now what happens? Given the limit of the resolution of the light microscope, the largest useful magnification, the level of magnification needed to increase the size of the smallest resolvable object to be visible with a light microscope can be determined. Or I can just detect specks of 0.2 millimeter. Alright, so it was 0.2 millimeter. Yeah, I was wondering what was the limit. So, or I can just detect 0.2 millimeter, while this microscope can go in theory to 0.2 micrometer. And consequently, the useful limit of the magnification is about 1000 times the numerical aperture of the objective lens. Alright, I get it. So most standard microscopes come with a 10x eyepiece and have an upper limit of 10,000x with oil immersion. A 15x eyepiece may be used with a good objectives to achieve a useful magnification of 1500. Any further magnification does not enable a person to see more detail. Alright, so they are saying this is the limit. Any further magnification means enlarging the structure or image doesn't means that you are going to see more detail, like you can see more detail than this. Indeed, a light microscope can be built to yield a final magnification of 10,000, but it would simply be magnifying a blur, right? They are saying beyond this 1500 times you can see, if you can go to tell 10,000 to see an enlarged image, but it will be blur. It wouldn't let you see inside more, right? So if you want to see a detail, just go till 1500. So only the electron microscope provides sufficient resolution to make a higher magnification useful. Yeah, so only electron microscope has this much resolution to distinguish between objects, and in turn the magnification becomes useful. Like magnification without resolution is nothing. You know what I'm saying, right? You watch a blur video at 144 pixel. However you enlarge it, you can enlarge it, but will it be functioning normally? No, you cannot see resolution. While if you watch 1080 pixel in your mobile, right, not in your LED or something, you can enlarge it and the quality won't be changing, right? Even if you enlarge it, you will see clearly the word as you saw without magnifying it, or you can say without zooming in. So magnification without resolution is nothing. Well, you can make a rough sketch of what the organism look like, but not the inside or not the details. So this was all the resolution topic, and let's go towards the types of microscope. Not the bright. Guys, I think we should study just the dark field microscope and leave the rest to a second day, right? We are tired. Or you can say I am tired. I'm actually sick. But I want to look at these diagrams. These are so nice. Alright, this is dark field microscopy, and we are going to see the these microscope. So the dark field microscope allows a viewer to observe a living unstained cell or organism by simply changing the way in which they are illuminated. Yeah, so they are saying how the illumination works changes how we see them. Unstained, guys, this is a big deal. Usually when we study things under the microscope, we stain them, because the microorganisms usually look the same as the background. It is more towards the transparent than being colorful. So how do you see a transparent thing? It's of course when you change the background into another color, then you can see that kind of invisible, or kind of colorless thing, cell or organism. It can be anything. So how do we change the illumination? A hollow cone of light is focused on the specimen in such a way that unreflected and unrefracted rays do not enter the object. Usually everything goes right, like the unreflected or unrefracted, like the light that didn't even pass through the specimen, it even goes into the objective lens, right? But in this dark field microscope, how do we change the illumination? We don't let the unreflected and unrefracted rays to enter the objective lens. Alright, what is this? Only light that has been reflected or refracted by specimen forms an image, right? The field surrounding the specimen appears black, right? Because there is no image for the background, because the light that, like, reflected or refracted into the background, we don't even capture them, and our eyes can only see things through light. If you take out the light that was reflected or refracted by some object, we cannot see that object. Even if it is a background, we see it as a black. Why do you when you close your eyes it appears black? Because your eyes aren't capturing light, while the object itself is brightly illuminated, right? The dark field microscope can reveal considerable internal structure in larger eukaryotic microorganisms, right? They are saying that we usually use this for what? Internal structure in the eukaryotic microorganisms. Guys, for example, yeast. Yeast is a eukaryotic microorganism, and it is a single cell. Like, you don't put the whole eukaryotic microorganism, like, well, we are eukaryotic as well. Do you put me under the microscope? I'm a human. Do you see me? No, we are talking about the cell. You take the cell from me and put it in the microscope, and there are organelles that we cannot see, so you can see that organelles, right? Figure 2.8ab, figure 2.8b. Right, let's go and see. Or should we change the or should we complete the paragraph? Then after that we should go. Yeah, we should complete the paragraph. It is also used to identify certain bacteria, like a thin and distinctly shaped distinctively shaped Treponema pallidum, the causative agent of syphilis. Oh, we know syphilis. What is syphilis is. Alright, so thin like and distinctly shaped. I think this is more transparent, so they are showing us extra clearly. So first, let's see the dark field microscope structure. Look at this Abbs condenser, right? Guys, look at this. So this is specimen. This is Abbs condenser. This is dark field stop. An objective. Oh guys, these are lenses, okay? Alright, so this is the collection of the lenses that we use. So what I'm not getting is, oh, okay. Okay, guys, look at this. This is the condenser and this is the source of the light, right? Dark field stop. There is a dark field here. Oh guys, this is way interesting. Then you guys can think of it. First you put a background that is of what color? That is of dark color, right? Because there is no light here, there is no light emitting from it, right? Because the light that is passing through this specimen should be something, so we leave this area as black, and then we uses the light from the sides, so like it makes a clear black background for the specimen. And when the light passes through the specimen through the condenser, because light doesn't go here and there because of the condenser, then it goes towards the specimen. Now specimen can reflect and refract the light, but there are some light that isn't being reflected or refracted, right? Like you can see this light is going straight into that direction. In case, look at this, they are not showing us clearly. Here, look at this. Like usually the light should go like this, right? Like this, there is a specimen, right? But in this case light has gone in that way and in that way, like oh my god, stop my drawing. Yeah, so it hasn't passed through the specimen, so it's neither reflected nor refracted, like it just went through it and passed through it for no reason. So this kind of light is not captured by the objective lens, and when it is not captured by the objective lens, the dark field, you can say the background actually works, and these kind of lights which is not even needed, the light which creates a light background, is just thrown out, and I don't know what this wheel is for, and let's see what they say below. The simplest way to convert a microscope to a dark field microscope is to place a dark field stop underneath. So first they are saying put this under. And how do you make this? I think this is how it looks, because this is why they have named them A. Well, B, the condenser lens system. The condenser then produce a hollow cone of light that is only light entering the objective comes from the specimen. Hollow light. What is hollow cone of light? I act like I understand, but I am not. Hollow act of, oh no. Hollow cone of light. A beam of light shaped like a cone, where the center of the cone is dark and only. Okay. I get it now. Guys, look. Wait. We have to do this. Oh yeah, I understand. Guys, look at this. The middle is completely empty. Look. The middle is completely empty. Just think about this light from all of the direction. They haven't shown the light from this area. Of course the light goes from three-dimensional. This is two-dimensional picture. If you think about this light, make it a sphere, so this middle area that is here will remain empty, while the light will be in circular shape all around this condenser, and that will pass through and make a cone, right? Right, I get it. You get it? Now I get it. But what is this thing? A wheel-like thing. Don't think we need it. A dark field stop. No. Why am I passing through it? We need to know what this is. Dark field stop. Opaque disc that blocks the direct light from passing through the microscope condenser. Okay. So to make a hollow light here, you need this dark field stopper that makes this light that is below here, stops it passing from going through in the middle. So it stops from going through the middle, while the sides one. Guys, I know you guys must have picked it quite quickly, but I'm not the smartest. So a hollow cone of light, so that only light entering the objective. Okay, that was that, and now we are going towards the interesting part. So they are saying this is dark field microscopy, and this is how the T pallidum looks like. Look at this syphilis. No, it's not syphilis, pallidum. It looks so vibrant, and you can see the shape, while the background is just darker. But did they told us to look at the diagram? 2.8 AB 2.8 A and B. Volvox and Spirogyra dark field microscope. Alright guys, so this is a eukaryotic cell, right? So I think Volvox is this rounder one, and the Spirogyra is this one. Give me a second, I need to know. I think Volvox is the rounder one. Spirogyra. Let me see the separate diagram. Yeah, here. Look at this, guys. We don't even need to search it. Volvox. This is how it looks like. While the Spirogyra. Yeah, it looks like this. Dark field. Sorry, I don't feel okay. Look, the background is so black, and they already stained it as well. Look at the spirals. I think that's why it's known as Spirogyra. I think the DNA. What is a spiral thing? What is the spiral thing in spiral gamma? Images isn't going to explain it. It's chloroplast, specifically arranged in a spiral shape within a cell. Oh, am I idiot or something? It definitely looks green. Green means chloroplast. Alright, we are done with this. Here they are explaining dark field microscopy, not the daughter colonies within the mature wall colony and the spiral chloroplast of the. Yeah, they are saying that look, we can see inside the wall as well, and the things that we see inside the wall is the daughter colonies, the things that are going to become a mature wall colonies. And the rest of the two diagrams are for tomorrow, or you can say another day, for phase contrast microscopy and the contrast and the interface, the differential interface contrast microscope, the fluorescence microscope, and yeah, things like that. So we are done with this, guys. Look at this, it's very interesting. So we do have questions in this chapter as well. Alright, so this is it for today, and we are ending the stream. Goodbye, good luck.